You guys said so well that I will not be able to make anything that you said better.
Okay, cool. So next question. This is maybe another kind of broader question, but I was hoping maybe Dr. Cotta, if you want to start off with for the answer. But so we kind of want to establish a workflow of what actually it means to incorporate genomic information into like clinical care for cancer patients. So maybe like, what is it, like, how does genomic information actually alter this timeline of care? Like, what is the role of a molecular tumor board, and like, how effective would you like say they are? Like, what is the role that they play in actually ensuring that the patient they benefit from this genomic data that's being generated?
Yes. This is a very good question. How oncologists decide what treatment for cancer patients? And this is really, maybe I'm not the right person, because I'm more on a maybe not non-traditional, I must say, non-traditional medical oncologist, maybe. But in general, I would say 80-90% of medical oncologists will follow something called guideline, like a textbook. And there's a list of drug. If you go through A, doesn't work, let's go B, doesn't work, let's go C, and so on. And nothing wrong with that. It's based on evidence that portion of patients may respond, and some patients do very, very well. But truthfully, it all depends on which cancer you're talking about. But let's say aggressive pancreatic cancer, unfortunately, most patients do not do well. But that's how patients are treated most of the time. I'm not saying that's correct or wrong. It's just what it is. I think in the future, if we can learn more about how we can use the genomic information in the future, I think what could be tried is to treat the patient based on available molecular information. Let's say if patient coming with a gene A, B, or C, and if we have a drug that targets those genes, let's try, and not in a one-by-one approach, because we know one-by-one approach has some limitation. History already knows that. We already know that from a lot of different clinical trials. But how about trying a combination approach and see how those patients do? The tricky thing about it, and this might not be jumping ahead, but the tricky thing is that when we do more and more genomic testing, let's say 400 gene panel, 500 gene panel, even the patient coming with the same histology, let's say pancreatic cancer, colon cancer, under the microscope, they may look very similar. If you're diving in a gene panel or gene testing, they all have a different molecular pattern. It's like a snowflakes. So in my clinic, under protocol, we have a good protocol to do that. In my clinic, most patients are getting different medications based on those gene information. But I think I will stop there. But I think that maybe the future, we don't know. We'll see how it goes. I think we have to do something different, that's for sure.
Yeah, great. Do either the other two panelists want to add anything to this? Their own kind of like understanding of what is the importance of doing this kind of testing? Like, how does it actually change the way we're treating cancer patients nowadays?
So in general, again, my background is different. But being involved in clinical trials and the ones we are designing right now. So for us, it's very important in the new era of cancer genomics, if you will, or precision medicine, is to take into account the mutational burden, take into account what are the mechanisms that are deregulated. It may not be a molecule A or B or C. So that would be ideal, but maybe a pathway, so a series of molecules that are deregulated and target these particular cancers with a particular alteration in a different way than the rest. This is what we call, to some extent, we call trial enrichment, so might not be perfect, but to give you an idea, so some of the molecules we're working with target one particular pathway, whatever that is, like a TMTOR. And so patients that will have mutations in one particular molecule, PIK3CA, in principle, we don't want them to be included because we have enough evidence based on animal models, based on lab work to support that these patients will not benefit. And so when we want to launch a trial, and we did that before, and we're doing now a second one for oral cancer prevention, for example, we want to be very targeted. So it's not that we want to leave people out, but the initial, so you really need to follow the evidence. And the evidence is that there is an enrichment for a group of patients that will respond better, then you go for that. It's more likely that the trial will succeed. You will learn why some of the patients still will not work, but at the same time, of course, you leave some out of this, but to some extent, you don't subject patients to a treatment they will not be, that you can predict most likely will not be successful. So there will be, you can say, would be unethical to include someone with a treatment only because it's approved, and you can do it. Yes, you can do it, but you know it's very unlikely to respond. So then you can explore something else. So that would be my take. So in terms of you can say the big population of scientists that work in cancer, our responsibility is precisely that, to identify, working closely with our colleagues in the clinic, really talk to each other and build on the information, so the genomic information they acquire, and this is what we are doing with Shumei, for example, and learn from that, develop animal models, develop cellular models, develop experimental models at the very end to test that concept. And at the end, it's much better to identify who will benefit based on these models, at least, who may not. At the end of the day, you do need to do the clinical trial. Let me give you one example that is completely different. So we completed a clinical trial using a drug very well known called metformin for prevention of oral cancer. And our expectation will be, based on our evidence, that this particular mechanism that will be, we can exploit by using, in this case, metformin to diminish the oral cancer, the oral premalignancy going into cancer. And when we did the initial study, our data were very clean, and animal models, beautiful, everything's great. We did a clinical trial, and turned out that the patients that benefit the most are those that are smokers, prior, current, or former smokers. And so to some extent, we did not expect that because our data have not been provided us. So right now, the next clinical trial will focus in that component, number one. Number two, we need to understand why this group benefits. We will not be subjecting people who don't have smoking history or current smokers, so they have different type of alterations, genomic alterations. They are copy number gains, basically complete different pattern of genomic alterations. But for those that have these complete different patterns, now we're developing animal models that will reflect that. You know, as we need to be responsible. We don't want to leave anyone out, if possible, but we do need to be responsive to help the physicians, in this case, identify the best patient population. And the same is true for many, many others. And building also what Dr. Kato Shumei was saying, for example, if you take lung cancer, so he can explain much better than myself. So the lung cancer, adenocarcinoma of the lung, is a very, very large population. In general, is one of the most frequent tobacco-induced cancers, and there's a repertoire of mutations, and they have a different distribution among the patients depending on the socioeconomic status, even racial differences. There are really clear differences. But right now we know that some drugs will only work when you have a mutation in one of these molecules, may not work forever, but will be responding in this particular case. And so basically what you do is to take one cancer called adenocarcinoma of the lung, and then you can parcel out in 1, 2, 3, 4, 5 different subtypes and identify who will be responding. This is what is really amazing is happening right now. And many of the new drugs that have been approved at the FDA right now, they are only for those patients that have mutations in, or for example, ALK or many others. That would be the concept. And the last concept that I wanted to bring is that you may not know what is the target, or at least you know molecule A, B, and C, and the typical one would be the BRCA mutations, for example, in breast cancer. You may not know, you don't have a drug to target BRCA, So people BRCA mutations, the people who have BRCA mutations are high risk developing usually metastatic, but breast cancer and also ovarian cancer and others. You cannot target that, but it's a new concept that emerged in our field. It's called synthetic lethality. So the idea is extremely simple. You have a gene A and a gene B, and if you target with the drug gene A, nothing happened to the cells. Target gene B, nothing happened. But if A is altered, now gene B is very sensitive. So if you target gene B when A is altered, oversimplifying, of course, then becomes very, very sensitive. And so that is really the concept is being applied right now for BRCA mutant patients in the use of different drugs. And Shumei will be the best one to explain the different treatments. But in the treatment plan, if you do have these mutations, then you are much more likely to respond to certain therapies that otherwise you would not be responding. Again, using the concept, you may not be target specifically BRCA, but you can use these so-called synthetic lethal interactions to make it happen. So that would be in a more pragmatic way how we approach develop treatment plans based on the genomic information, based on, for example, tobacco use, which may not be genomic driven. So just to give you an idea of some, and Shumei can also elaborate in terms of immune therapies. It's very dependent, not completely, but depending on the status of some molecules like PD-L1, you may choose different treatments. So it's wonderful for my colleagues, they are physicians because they make the decisions. But again, so our role is to work close with them to identify all of these, we call vulnerabilities of the system. So something that may not be obvious, it's not going to be a gene that's mutated, hence you do something, but you can build on that. So you can harness the information to target that.
Oh, wonderful. Thank you so much for sharing those examples. I think it really speaks to the complexity of the issue and how much of the data we need to parse through them, like before we can be really conclusive about any of our results.
Yes, I want to, sorry, I may have mentioned something that is negative, but yes, I echo Silvio. There has been a lot of progress. And definitely we, like a lung cancer example and the PARP inhibitor example, definitely there's a huge advancement, but still, I think we can do better. So that's, I think what we want to do is moving beyond what we have right now. So just want to add.
Okay, cool. So next question. So what are the biomedical challenges with scaling up precision medicine so that it is an accessible tool? Because, Dr. Gutkin, I know your lab works is to actually identify the markers and for like this actual cancer stratification. And so I think of it as like two things, like there's the healthcare delivery system side, and then there's the side, like the side that actually generates like the data that actually gets applied by, clinicians. So I guess looking more at like the second side, like what are the challenges of actually like scaling up precision medicine? So it's not just like, okay, we're going to do all of this work for one patient and then that was a really exhaustive kind of process, so we can't just freely do it for another patient.
I think that my colleagues, Shumei and Sandra, you guys are better than myself to respond to that.
Well, that's a really tough question in terms of scaling up. I think that is the $1 million, maybe multi-million-dollar question. I think, in terms of the integration of, and I don't know if this is what you're getting at, but the integration of electronic health records and bioinformatic tools in terms of disseminating and automating information quickly into the charts for patients so that it's at least available for clinicians to act upon, I think is obviously really important. I mean, we studied a community-based healthcare system up in Palo Alto that was trying to integrate and create a biobank that was going to be linked to their electronic health record system. And it was really interesting because one of the biggest challenges as they were trying to launch was just explaining this to patients and getting their buy-in in terms of allowing for that kind of linkage to happen. And also, you know, it really, again, I'm going to make the point that it really was, it was a different perspective from different populations in terms of the value of this and the investment in this, and also what was perceived by some as, you know, all of this data about me, intimate data, is going to be out there. And yes, it has the potential for coming back to me in a beneficial way in terms of clinical care, but it also seems to me to be highly risky. And so I think that to mitigate some of those concerns, there needs to be a lot of transparency in terms of how these systems are being built, who has access to them, what is actually the return on investment for a population that might feel vulnerable. I think that's a really important aspect in terms of scaling up and getting public buy-in.
Yes, that's a very good point from Dr. Lee. There's a lot of issues actually. I don't know if I can cover. But if I may comment, you know, I think in a cancer field, when it comes to, you know, getting, let's say, FDA approved medication, the gold standard was that you need to do the phase 3 study, you know, big study, maybe 500 patient versus 500 patient. You give a standard chemo in one side and you give a new medication and randomize and follow up the result and open up the result and say, if the new medication is working, approve. We'll move on from there. But one of the challenge these days is that, let's say, you know, give an example, a fusion, broken gene called NTRK, NTRK fusion. Can you do that study? The answer is no. It's only present in less than 0.5%. So we cannot do such a large study to prove in a phase 3 randomization. But luckily, I think, you know, FDA has been very flexible these days, that even with a small number of a patient, without the randomization, let's say 120, 200 patients data, they give approval. And they even give an approval in any histology, meaning, you know, usually the drug approval has been based on a cancer type. Okay, breast cancer, this drug. Lung cancer, this drug. FDA has been very, you know, open these days. NTRK, if you have NTRK fusion, any cancer, it's approved. Even the immunotherapy, as Dr. Silvio mentioned, you know, high, you know, tumor mutation burden, MSI. If you have it, doesn't matter, it's approved. But I think we'll learn more and more after the approval, you know, how we can wisely use this information. But I think, again, one of the challenge, especially in cancer field, is that the gold standard, the textbook way of getting drugs into market or patient has been a large randomization, which takes time, which takes a lot of effort. And but people are realizing that may not be the best way. So we'll see how things go.
Cool. Yeah, that kind of leads into this next question. So does clinical genomics bridge or extend the gap between physician and patient knowledge? Because I think we have this really rich set of genomic data, and in general, patients do place a lot of trust in their physicians because the physicians have this kind of really large responsibility to actually interpret the data. The patients don't always know, like, okay, is that a broken bone, or like, is this cancer? So I guess, like, does generating that extra layer of what should be objective kind of quantitative knowledge, does that actually add clarity for the patients, or does it kind of actually blur things more? And does that mean that the physician should, like, what are the new responsibilities that a physician may have to take on with the incorporation of these technologies?
I think if I make, you know, comment, and please tip me in, Dr. Lee and Silvio. You know, patient who see the patient, you know, cancer patient like myself, I'm far from a scientist. You know, you don't know everything. My knowledge is limited, so definitely we need a support from you know, scientists like here. So definitely active communication and learning is necessary. But one of a potential tool, I think, is as you mentioned earlier, something like a tumor board, like specifically for molecular. So there, we can sit down with a group and discuss what is this gene is potentially doing. How can we target that? Do we have a clinical trial so that each physician do not get all the full of those massive information and get burned out, because impossible is for us to understand everything. But I think as a group, hopefully we can tackle this.
Yeah, I would just echo the challenges that that were just mentioned in terms of relying on physicians. I mean, they're really doing the heavy lifting, because we can see time and time again, patients look to their physicians for guidance. They're the most trusted messengers, particularly for technologies and approaches that are new. And so there's a lot of pressure on physicians to be able to kind of be those knowledge brokers for patients, and to be able to communicate in a way that is effective, right? So genetic literacy among the public, I mean, even among experts, it's a challenge, but for the public, it's particularly difficult. And so, trying to, as we move into the genomic era, for example, the idea that many genes have a small effect on a particular outcome, that's really hard for the public to understand. And even this notion of risk is a difficult concept. So I think that there needs to be not only help for physicians, but also perhaps a new cadre of providers that can be a conduit between physicians and patients. So we normally rely on genetic counselors, for example, to relay information in a way that's comprehensible to the public. But as we know, there's a real shortage for genetic counselors, particularly genetic counselors from diverse backgrounds. And so, is there a time, as we're ramping up in precision medicine, should we also be investing in a workforce that can help deploy the information effectively to the public? I think that's probably the case. And we don't want to play catch up where we're constantly trying to build something that we should have been building in parallel, perhaps, with the science.
So we had precisely this discussion today with an opportunity to collect a huge amount of data from the entire UC system, and where that will be kept. And I mean, there's a lot of information, certainly. This is not a gigabyte or terabyte. These are just amazing amount of information, and confidentiality. There are many issues. So what we discuss as well, aligned with the comments from my colleagues, so that maybe it's time to start developing what we call genomics navigators, people who can translate in the right language, in the right person, for the right culture. They can translate all of this wealth of information. So because, again, so you may identify so many changes, and some of these may not really be, I wouldn't say relevant, they're all relevant, but maybe limited impact. And so when speaking about some of our patients here, so when they receive a report from some of these companies that are even running 500 genes, or can be the entire genome, which we can do these days for not too much money, then they receive a report, and the report is just absolutely impossible to decipher in any language, English included, okay? Forget about other languages. So it's very difficult. So that is where you need the physician, but also someone in between. Especially, so that would be similar to genetic counseling. In this case, so to take not only the genetics, but we call omics navigator, someone that can capture all of this and try to work with the patients and explain what from this is really relevant, what is not. And so the concept we are beginning to use is whether something is actionable. So whether it's something that we can do something about it, and whether if we do something, we have evidence that can benefit the patient as well. It's not overwhelming with gazillion mutations, which we love, by the way, to identify them, but then there's not much you can do about it. And that brings one concept that I will put forward, is the ethics of precisely defining we can define risk in some cases. We can define very clearly risks with some mutations, and there's nothing you can do about it. So is this ethical to tell someone you have a mutation, very likely you will develop metastasis in X number of years, very, very likely with, I won't say 100%, but very, very likely, and yet there's nothing you can do about it. So that brings a lot of issues that some of my colleagues working in one particular type of cancer in which that happens, they always struggle with that. So the communication is central, communication in a way that the person can understand fully, but although at the same time, the ethics of it is not actionable, that creates a little bit of a problem. So this did not exist before. Now we have that information. So I'm bringing up something that is a little bit different, but this is the extreme in which you do have information is very valuable, predicting the patient will metastasize, for example, and yet there's nothing you can do about it. I'm not making those decisions, so my mice don't complain. Yeah.
Oh, that's a really valuable point. I think it actually goes right into the next question I had. So, what are the implications of direct-to-consumer cancer genetic testing? Because, like you just said, Silvio, like the role of or actually all of the panelists have said, like the role of the physician is very paramount. Like they are often considered like the kind of the person to trust, like the main person to actually relay all of this oftentimes convoluted kind of medical information to their patient, and the patient places a lot of trust in their physician to kind of guide them through this oftentimes very challenging process of dealing with cancer. So direct-to-consumer testing actually kind of, I feel like my impression is that it kind of removes the physician from the picture, and it kind of puts it, the goal is maybe to put more power in the hands of the patient themselves and actually allow the patient to kind of take ownership of their own genetic information in a way. So for example, like 23andMe it was a really popular, it's been a popular choice for people. They can just, do the swab test and like get back this list of like really maybe not interpretable for them, maybe like listed like risks, or like they get this like, oh, you have a certain % chance of being like this or this. So I guess what is your understanding of like the implications of this kind of testing that kind of removes the physician from the picture? And is there a place for it? Like, do you think it's valuable to be able to empower the patient, or should we try to do more to kind of build that relationship?
So maybe I'll just start. We actually did an ethnographic study of 23andMe about 5 years ago and their consumers. And it's fascinating because I think as you say, sociologically, it's very interesting in this country where, we went from an era where as you say, healthcare provider was the gatekeeper, and you actually had to have a reason for getting a genetic test in the sense that it was for an actionable condition, that you had a family history, that you really had to justify having access to that technology. And we've swung the pendulum completely the other direction. And that now, the kind of narrative around right to know, you own your data, you should have access unfettered by any gatekeeper. And 23andMe and companies like 23andMe have really kind of ushered this new era into existence to a large part. I think what's interesting is this kind of conflation between patient and consumer, and that's a really important distinction. And I think that companies like 23andMe have conflated them so that now you bundle really important cancer test results with your ability to smell a particular thing, right? So, or this kind of recreational genetics. So when you do that, it actually becomes, I think, more confusing for the public. They don't know the weight of the evidence behind these tests. They also, it's very confusing because it's oftentimes the limitations of the test and what they do in terms of follow-up is unclear, because you're just a consumer, you got your test results, you don't have your physician or somebody to help guide you. And so I think it can be actually quite it can be quite confusing and perhaps even kind of definitely ethically fraught, but can be quite harmful if this information isn't kind of scaffolded with all of the information that you would want a person to have. Maybe I'll just stop there, and I'm interested to hear what my colleagues think about this approach.
Yes, that's a very good point by Dr. Lee. I'll tell you from my, my personal experience, I guess. So again, comprehensive genomic testing, especially with next-gen sequencing, getting a lot popular. And nowadays, as you know, you can do it through blood. You can even detect the DNA shedding from a cancer, from a simple blood test. So, so I'll give you one example. 3 days ago, my patient emailed me and say, hey, I got this blood result, full of report, like a 50-page long. Say, what can I do? So people are coming in with all those information, not directed from you, but by patient. And I actually, like to have more information, so that's good. But again, it comes to the burden, meaning not everyone can interpret, including myself, the full picture of those information. And what is also confusing, it could be confusing for a patient, is that it's hard to interpret, of course, even for us, for anyone. One time patient told me, hey, I have a gene called TP53, which is a very common, more than 50% of cancer have this. It's a tumor suppressor. It has a lot of function. And patients say, hey, I found in Google, green tea works. Can you prescribe me a high dose green tea? I said, yeah, I mean, if you look at the literature, there's always some association, but I don't want to do that. So you need to deal with those, conversation, which is okay. I enjoy it. But bottom line, I think we need to have a good system who can arrange those testing or interpret and give a recommendation in a systemic way. But when it comes to getting more testing, I'm for to it. More, we need to learn more about the cancer, because we're not curing. So bottom line, I'm okay with more testing.
So that there are at least two different ways to see it. So one is precisely, so the more information you have, you may not be able to have an impact right now, but if as a community we collect a lot of information, call it big data, in terms of genetic variations or genetic alterations in cancer, for example, and you collect all of this data, so patterns will emerge. When you have that's, I wouldn't say is a given, but very likely the more information you have, the more information in the future can be actionable, so you can do something about it. Maybe not this particular p53 mutation, having p53 mutation, but maybe a subset, they will respond to something better or worse. So that will come with some time. Very, very important point that was made at the beginning. So quite often, data sets do not include a diverse group of people. So this is really damaging. So most of the underrepresented minorities, they are not that in the data sets, even for our own work, they are really underrepresented. And that is basically you can say that the benefit of whatever, even 5-10 years from now, all of the aggregated data that we are paying for one way or another as a society may only benefit the subsets unless we make sure that everybody is included. Going into the other extreme, there are two classical ways, at least for cancer. So one issue will be the associations based on mutations. These are somatic mutations. Your body have a normal gene, your cancer has a gene that is altered. And that is, we can understand and embrace that. And that is where most of the action is. There's a big push right now also to have what is called germline variants. Basically, that means that your entire body, so one gene like A, your gene would be a little bit different from mine in some way. So it's not a cancer gene, but these are the ones that can predispose me or you or whoever to be affected in the future for any disease, including cancer. So the germline variants are much more complicated. At the very end, this is what we wish to know, because we can predict risk. And if we can predict risk, we can intervene early and eliminate the risk. Or maybe some people will be at risk from tobacco, some people may be at risk from living close to the highway and the polycyclic molecules coming from the gasoline, so would be effective. So there are many ways to do that. That is the most difficult because really most of these alterations have very limited impact. So huge studies have been conducted. They're additive, can be additive or can be synergistic. So alterations in two genes, these are normal genes, may predispose a lot and few others no. That brings a lot of the issue of the environment interactions, so gene environment interactions. And certainly the only way is in confidentiality. So you don't want to disclose everything. We would like to have everything from you, but we cannot, we shouldn't. And so this was, so for us, thinking out loud for 20 years from now, we have the opportunity to collect all of this information that we can predict risk with the 23andMe, with many markers, which now you can do. You would like to be very robust. You would like to really cover everybody, but also have enough information about gene environment and familiar cases, exposure, etc. So that would be the most difficult, and again, very difficult to capture everybody, provide that opportunity to everybody. That would be even more difficult than currently what we are doing for these panels that are mutated in cancer. So when you go to the implication for direct consumer, you need to think about cancer genetic testing. You can just make it broadly genetic testing and identifying that those variants that can help you identify risk. I think that would be the most difficult part, but looking forward in a few years from now, maybe 5, 10, that would be probably what we like to be, and will, there would be a lot of even more complicated issues regarding ethics and making sure that everyone has access to. So this would be for you guys, so that are, you will see that we may be already doing something else.
Well, thank you so much. Yeah, I think that's a really compelling topic. So we're almost done, but I think we have time for maybe 1 or 2 questions. So if you guys have any questions, feel free to raise your hand and ask a panelist. I think we actually have 1 question. Oh no, sorry. Okay, so yeah, if you guys have any questions, just raise your hand and ask, and go ahead, or just speak up. Yeah. Gordon.
Hi, I actually had a question for Dr. Lee. So I think recently there's been this concern about equitable precision medicine and ensuring that data sets are heterogeneous and reflect the diversity of the population. So on the road to equitable precision medicine, what do you see as kind of the strengths and concerns of using diverse data sets, such as the NIH All of Us research program? Oh, I think you're muted.
Thank you. It's great that you mentioned the All of Us research program, because that's certainly an initiative that is trying to address some of the issues around diversity. What is your question, what are the harms or what are the concerns when we're in this kind of what we hope is an intermediary state before we are able to obtain inclusive references? Is that?
Yeah, so I think the main thing was like, as an informatics person, certainly it's exciting to have access to such diverse data sets. But from an ethics standpoint, will simply more diverse data sets help us answer those big questions in an equitable manner? And are there any unique ethical concerns that arise from using such population level data?
Yeah, well, so there's a whole host of things that we could talk about, but certainly this issue around goals of equity. I mean, there's this stage where you're trying to do kind of more basic science and understand what the important genetic variants are, and certainly having an enriched reference set will help us. But in terms of the translation, kind of further down the line, whether or not that translates into equitable therapies and better health for certain populations, I think that's where, at least in our research, and we are constantly interviewing and talking with participants of various precision medicine initiatives. There is some healthy skepticism about the ability to reap the benefits equitably across groups in terms of this investment. And so, and part of that is because of our healthcare system, frankly. And oftentimes we try to decouple research participation from the actual healthcare that's delivered. And I think thinking about ethics in terms of an ethics of care, rather than just thinking about research ethics, but thinking about it in terms of the whole translational pipeline, is really a move, I think, in the right direction in terms of understanding what actually is delivered in terms of addressing health disparities, because that was the rhetoric around, for example, funding the All of Us research program is addressing health disparities. And so being able to attend to the healthcare inequities that we have, I think are really going to be important.
Thank you. Yeah, I think that makes a lot of sense, especially because we want to make sure that all of those claims, like they're not just used for good marketing, but that they actually translate into a real tangible benefit. So thank you.
Mm-hmm. Yeah, thanks so much, Dr. Gutkin. Thank you so much for coming here. Yeah, I guess if maybe the panelists have maybe time for one more question, we can ask if anybody has one, or if not, we can call it the end. Anybody have any last question to ask?
I don't want to go over time if people have places to go, but I did have a question regarding DTC. So when it comes to having all of this new information available to the public with relatively like short amount of money and time, how will we go about with promoting education for what is available to us, especially with new like medical like information about ourselves?
Do you mean through DTC?
More about, yeah, about DTC. So if this is available to us, but one of the main issues is that there's a discrepancy on interpreting what this actually means for our bodies and how to interpret it. How would we go about educating the public in order to make this more of a viable method?
I was going to ask Dr. Cato if he wanted to answer, but no, that's a great question. I think it's a real concern because, as I mentioned, you know, this the pendulum shift that 23andMe and other companies have pushed us towards is this idea that if you have the information, somehow you're empowered. But as we know, just having a report doesn't do you much good unless you're able to interpret it. And I think there's going to be a tremendous need for figuring out how to educate the public about the limitations and the possibilities of the results. You know, so far, it seems that this is largely coming from the companies themselves, frankly. I mean, if you look at the various companies, they have extensive modules on how to interpret their information. You know, as an ethicist, I'm worried about where that source of information and whether or not it's an unbiased set of materials. And so I think we're still kind of searching actually for good sources of information to interpret the data that we're getting back. Because really it's in some ways it's just data. It's not information until you can actually use it in an effective way. So that's the challenge.
Thank you.
Great. Thank you so much again to everyone for coming today. I hope you had a great time learning more about this really controversial issue, and hopefully we gained some clarity through our discussion today. So thank you again.
Great. Take care.
Thanks so much, Dr. Lee.
Sure.
Okay.
Bye.